Well, good evening, everybody, and welcome to our seminar today on tuberculosis. My name's Dr. Andrew Burke. I'm an infectious diseases and respiratory physician here in Brisbane. I'm also affiliated with CRE Respond, which is with the University of Queensland Centre for Clinical Research. So we do a respiratory infection seminar once a year as part of our educational series, and so I'm really excited with the three speakers we have this evening. Before I go further, I'd just like to acknowledge the traditional owners and the custodians of the lands on which we're meeting, and we pay our respect to their ancestors and their descendants who continue cultural and spiritual connections to the country, and we recognise their valuable contributions to society. So we have, in the area of tuberculosis, I should say, we've had a lot of interesting clinical trials, landmark clinical trials over the last 5 or 10 years, which are really changing how we treat TB, and certainly in this forum we've presented those in the past, and I know our third speaker, Gail, is convening a symposium on clinical trials next Tuesday, if people are interested. But I thought I'd choose 3 topics which may be a little bit, maybe left of field is not the right way, but a little bit different, a different angle on tuberculosis treatment and how we approach it. And so they're 3 very different speakers, and I've had the great fortune to hear them all speak in different forums, and so very grateful that they're speaking today. Our first speaker, Professor Shin first, then Dr. Gail Cross, and then Professor McBride, and we'll aim to finish at 7:30 Brisbane time. So our first speaker gives me great pleasure to welcome Professor Gook Shin, who is currently Professor and Chair of the Department of Pharmacology and Clinical Pharmacology and Founding Director of the Pharmacogenomics Research Group at Inje University in Busan, Korea. He's also Director of the Centre for Personalised Precision Medicine of Tuberculosis at the University College of Medicine. He's had a number of very interesting roles, and he's had many esteemed roles in academia and also in industry, and he's CEO of a bioventure in drug development and personalised precision medicine called SPMED Co. After he received his PhD in pharmacology, he's done a fellowship at Georgetown University for 2 years in the United States, and he's published over 350 papers. And so I had the great privilege of meeting Professor Shin in Taiwan in May and heard him speak on a similar topic. And we're about to start. We've been trying our hand at TB therapeutic drug monitoring for a few years now in Australia. And we're in the process of starting to write a TDM guidance document for Australia and New Zealand. And so this talk will be very relevant to what we're aiming to achieve here in Australia. Thank you, Professor Shin. I'll hand over to you, and I'll advance the slides when you ask me to.
Thank you, Dr. Andrew. It is my pleasure to be in this sharing the Respond seminar as a speaker. It is my pleasure to introduce some of the TDM and also model informed precision dosing concept to colleagues in Australia. The TDM, the therapeutic drug monitoring, is a tool to used to integrate the pharmacokinetic and pharmacokinetic knowledge to optimize and personalize drug therapy. As you can see here, we do measure the drug concentration, and we need to keep the concentration within the therapeutic target. If this is going to be within a therapeutic range, that has a higher the probability to get the therapeutic success, or just below, it means had a higher chance to get the therapeutic failure, or just going to higher level than the range, there is a higher chance to get the toxicity. So in the conventional approach, we do titrate the dose to keep the therapeutic target range in the case of TDM based approach. And now we do have something the more complicated PK or PK/PD modeling. Then just we do just call model the informed precision dosing to target the optimum dose based on the AUC or something AUC or MIC. Although it is not well established so far, I just interested in all those concepts in the later. And for us to do this one, we needed a population PK model for TDM based dose adjustment. Maybe we can do also get the PBPK, P0 based PPK modeling as well as one of the alternate way methodology. Why we need the concentration control the dose precision dosing, because as you can see here, we have the wide individual variation in the antituberculosis concentration. You can see here all the drugs you can see have showing several tens and the interval variation and the key, given the drugs. So what about the usefulness of the TDM in the anti-TB drug medication? This is the optimization therapy to ensure or improve success in a specific clinical setting, or manage the PK drug interaction, or drug disease interaction, or new anti-TB drug formulation. In the case, what about the David's formulation may cause different the concentration or PK profile. Therefore, that could be helpful. Or in the influence of dietary contents or anti-ARS drugs on the bioavailability. So therefore, when those patients were the exposed to those situations, we may do. And also, one most important issue is also good to just keep the compliance of the patient, drug compliance, as well as I just told you, dose adjustment to reach the drug packet concentration. For those, the purpose, we do just measure drug concentration as a condition to titrate for the optimum the concentration from the prediction of the optimum dose regimen. We just call precision dosing. So for us to do this one, to target, we need the target the concentration. As you can see here, we may do have something the known therapeutic target the range of the concentration by something the groups. And although it's not very popular, but this kind of the target range is used this Cmax or 2 hour and 6 hour fixed the concentration are used for this one. But in this slide you can see here, as well as this you can use the AUC as index. But also, using the AUC and the Cmax, we do measure, we can do just use the AUC or MIC together with the consideration of the drug sensitivity to, the TB bacilli. Therefore, you can do have the many drugs have something target the AUC or MIC concentration to have the better or the response, as you can see here. Here and you should know the most of the this AUC MIC data are mainly just determined by the kind of the in the in vitro study named the Hollow-5 infectious model based approach, because we have the very limited data for the AUC and the related drug response based on the MIC in the clinical setting. So you can that is a big issue. So why you just the interest in the drug concentration? Because you can see here in the concentration is related to the drug response. So as you can see, with some clinical data published, the AUC or Cmax seem to be just correlated with such the outcome, the response, or some toxicity, or according to those issues. So therefore, this is reason why we are interested to the concentration in the many infectious drugs. As well, also we can just collect many information regarding on those for the rifampin, pyrazinamide, isoniazid, something others in relation to the drug concentration, educate low in the high drug concentration, how it is related to something clinical response. I not just talk to all those issues because of a limited time. And in this case, in the how we can do, the make the overall scheme for the model the precision dosing based on the TDM. And you can see here, if the patient have, and then we just collect one or two, the minimum blood sampling, then measure and the concentration. Through this, we do just have something the pharmacokinetic analysis. In general, in the PK analysis, we cannot do that only one or two concentration. In this case, if we just including something model as a prior information, we can use something the PK parameter estimation based with using kind of some methodology like the Bayesian approach. Through this, in the we do just the estimate the PK parameter of the individual patient. With the consider the MIC, we can do just evaluate whether it's the therapeutic target, or in that case, if unnecessary, we may readjust the dose and then just the circulate this the process. First to go this one, how we can do just to initial dose. The initial dose, as you may know, well know, the initial dose and the dose adjustment with using patient progressing, we need something the prior information for the PK or PK/PD data. So that is model. What we can use is population pharmacokinetics or pharmacokinetic model, or maybe we can use the physiology-based PK/PD model, although it is not used so far at the moment in the world. But this, the future coming potential model can be implemented in this, the TDM-based dose adjustment or dose estimation in the given individual patient for the MIPD. So for us to do this, we just have something, the 7-year project for the personal precision medicine of tuberculosis that is funded by the Korean government as a project of the medical research center for the promotion of the Beijing medical research in the country. So if I just interested in the overview of the R&D strategy, we do have the clinical cohort, the targeted 5,000 population of the TB patient. At the moment, we have the 4,000 cohort. From this, we do just collect many information, including clinical information, as well as the sampling of the blood for the drug measure, drug concentration measurement, or some the genetics, or the using for some other purpose like the metabolomics or some the immune profile, whatever. And also, together with those, and we do having, we are going to develop this pharmacokinetic, pharmacodynamic PK/PD modeling model, as I just told you, such as the PPK model and PPK model. PPK model is going to be just, developed initially via in vitro ADME data, then just ADME or some other prior, PK data. Through this, we do just develop, then just validate for the, the model with using the clinical data we just collect. Of course, clinical data will be used for population PK/PD modeling, and those model can be used for the predictive algorithm for the initial dose and the dose adjustment, as I told you just before. So all those are just, we do have the, collecting the workstation. There is a web-based workstation in which you do just manage all concentration or manage or just, TDM-based, the semi-automated PK analysis or even the consulting or just access to the data of the individual doctors who are just involved in our project together. So those, the approach that could be used for the precision pharmacotherapy for the future in the tuberculosis in the future. And today, and we are just much more just talk about this in the TDM and the PK model. In a couple of slides, I just introduced a little bit the HALO-5 infection model. For this, at the first, we need a good bioassay. So therefore, we just developed the simultaneous assay for the all anti-TB drugs. Over the 20 anti-TB drugs medication, which is under the prescription in Korea at least. And this is the LC mass chromatogram covering many TB drugs. You can see here, we do just developed the simultaneous assay for all those 24 drugs and one metabolite. Also added proteaminide at the moment. As I just told you, we have those, and then we also, developed for the potential use of DBS, dried blood spot, and also used and validate those methodology and covering the most of the anti-drug drugs. And from this, we do just get the drug concentration because in the TDM based our code, we do just collect one samples or maybe sometimes repeat sampling may get just one or two sample concentration from the one individual patient in general. So from this, this is all symbols of drug concentration of individual patient. We just collect in the different time point. From this, as you can see, they show the wide variation. But we can see what about the concentration in the given time point. What about it is belong to the range or not? But this is a collective concentration that will be used for the the dose adjustment and in the following process. Okay, so through this we do see whether the target or if the concentration not just within target, we just made the concentration control dose that based on the such as the model informed or the Bayesian with using the model with using the Bayesian the approach to estimate the pharmacokinetic parameter based on the prior information such as PAPK, as I told you before. Okay, that's the process. If you just compare from now I just interest a little bit just how the established the semi-automated the TDM systems. Therefore we are if it's the interest a little bit just the conventional versus the semi-automated TDM. So as you can see, the conventional approach we need get the charge doctors just request the TDM from the sampling of the drug consent the blood level blood samples. Then just we do get the clinical information as well as the drug concentration measurement with using mass, and then we do just the get something the estimate the PK based on the supported by clinical pharmacology and or predict with or with using some program, and then just go to the something the interpretation or give the report and then come back to the child doc. This is something the process that we need several steps, but through the semi-automated process we can do reduce such the steps to be more simplified and the more and the speedy and the process. If you compare the conventional versus MIPD based TDM such as semi-automated method is usually the conventional TDM do has that just direct implement you just see the Cmax or C12. The interpretation is a little bit easier because we just check whether the within concentration or not. But it is if you just titrate only just really using the concentration, it is take the time, the labors, but also that should be easier if it is going to be the state concentration really easy to be used by the clinicians, and that will take time and some frequent sampling, repeated sampling necessary. There's some the cons. And MIT based TDM with using some these the system is more rapid efficient, or we can include the covariate of the individual patient or PKPD model, not only in the concentration, but also we can estimate the AUC or Cmax or the MIC if we have the data. Or just limited sampling is okay, and also flexible time sampling is good because we are to estimate PK parameter. Therefore, we don't need to fix any specific time of the sampling like 2 hours or 6 hours, and also not necessary to have steady state at the ways, but we need a complicated patient forecasting tools. We need the prior information, and so also we also the person should know the expertise in the such the software like the PKPD, non-MEM or non-NIX or the R such as. So the the Bayesian method is a little bit just the not very complicated, but very good tools for us to the one samplings. In the conventionally, we do if we just estimate the PK parameter, we need to repeat the sampling, dense sampling, then we do measure the PK parameter. In the case the Bayesian approach, we do have the one concentration because we have the prior information. Then through this get the maximum probability the PK parameter individual estimated by the condition. That condition is measured the concentration of that patient from the clinical characteristic as well as dose regimen. So that could be good to estimate the PK parameter without having such dense concentration. So this one, as I told we need the process concentration character, the forecasting software we need, and the estimation, PKPD targeting, and the simulation, and the measure, and the interpretation, go back. And so as you can see, this is a process is a little bit complicated and also not just familiar to the many of the clinicians, or not everybody can do them, and therefore we try to develop something that the automated systems. So there is the composition of our semi-automated TDM based consultation. We just have the smart ID orchestration CPMTB. We do have the orchestrating the Amazon Web Service, and we do have the prior information, including PAPPK, PPK, clinical data, individual therapy target, MIC, distribution, the major drug concentration, etc. As well, we do use the NumMem R and R-Studio Java for coding and access, and we do have the web-based data entering systems, multi-use in the clinical side, the investigator or PK or TDM specialist, then just review then report to the child doctor. We secure the access to the own patient. This may make the decrease the burden of the human resources, and therefore at least we can reduce number of the TDM consultation expert who are involved in the TDM consultation process. So I just showed something, the example, the website snapshot of the web website of our the PMTB. In the starting, we do have the collect CPMTB code, and then we can just select the individual patient targeted, and then from this we can see all the information, the ID information of the patient, the demographic data, and other information, and the anti-TB medication history, or lab results are just available in the website, because just in our on the individual site they already uploaded those information in our the CPMTB. Okay, then just medication, please go. I just go past the next, the anti-TB drug medication, the drug concentration as well, and or adverse event or genetic information as well. So the next, please. Please go. We have limited time. The automate those, then we estimate the predict and estimate whether the predict the concentration is in the given individual patient. Next, please. Okay, then go. I just go left the place. Next, please. And this time course, and then we can simulate from the in the different dose regimen to may get target concentration. So through this, we can predict dose to get the target concentration as well. In our system also, because target concentration not always very good, and also not very well working, and therefore also we just give the information to our the site doctor for them see what about the position of the concentration, what they measure from the population data what have. It means what about it is very higher concentration compared to the others on the and too much lower on the given position. There is also good information for the doctor to catch up the situation and for them to just have the clinical decision for the adjustment. And from this we just give a result like this. Next. Next please. Sorry, go ahead. And this is the sheet for the report, including the information, dose, concentration, prediction, time course as well. Also we give the information like this, because the clinicians they have limited information for this. The parameter or the knowledge. So therefore, we just try to give something information together with reference. Next, please. We need a model for this MIPD based constellation. Please, next thing. Okay, I just go rapidly. The PAPPK model, how we can do. I just showed some example. Next, please. Because we have the many concentration data, we can do with the PAPPK. And also in vitro data we have as well. So we can do develop the PPK modeling. The both in the model can be used. The next, please. So this is one approach for the PAPK modeling. In the isonide, we have the huge data set, and from this we can see NAT2 genotype is one covariate or random function seems to have some trend to just showing a little bit difference as well. Food effect are available. So therefore, we are just going to develop the model including those covariate. Next, please. And for this modeling, we just get this, the metric plot or something to just look for the covariate the and then we develop the model like the generalized model. This model can be used for the the Bayesian approach based or TDM for dose adjustment or it's initial dose estimation. Next, please. Next. The dose was validated. It's a model validation for the the model already developed. I'll just go next. It is not your interest. Go next, please. And this one model validation method and how much our updated model is better than the model developed by the prior the literature that you see showing. Next. And from this, we can do estimate the initial dose based on the the NAT genotype, and we can just data is well just matched with the data, known just information in the global level. So anyway, we can use like this as an example. Next. Okay, and so next, please. According to the genotype, and through this we can have some case. Example, through this, because the NAT genotype is too ultra slow accelerate, and we need, the adjust the dose to the much lower, the regimen compared to the usual person. Next, please. Okay, and next here. Yes, okay, simulation methodology. And or just in the PPK model, this is more complicated, but I can say we together with the prior PK in the animal or something human data, but also the in vitro data you can do just include every covariate like the the nanohepatic dysfunction or whatever the genetics genotype of the individual or some drug interaction. That is really good methodology, but need huge effort to go. And so far, this is not just be used for the routine TDM based doses at the moment, but this can be used in the picture as a good alternative for the PAPK. Next, please. Okay, this is the methodology how we can develop the the model PPK model. We do have the in vitro data like enzyme kinetics, drug interaction data, or pharmacogenetics data. Pharmacogenetic data for the really using the expressed enzyme for the wild type and the mutant type. We can do just the use those in recombinant enzyme and seeing the the enzyme kinetics in those different one that could be used for covering those the such covariate in the the model. The next, please. So through this, we just can develop something model like the PAPK, but also different type of the model. So if you just have the model, we can do just predict the exposure like the Cmax issue. Compared to the control, just the TB patient, if you just add the DDI or pharmacogenetic together, or just the liver cirrhosis, the combined all together, or what the AUC exposure is almost the 6-fold high exposure is expected. That could be used for the the initial dose estimation, or that just the PK parameter can be used for the the Bayesian approach in the future as needed. Okay, next, please. Okay, and the issue is at the moment, if you just wanted to get more just the complicated modeling for the precision dosing, really using the MPK-PD index as a target, not only the fixed drug concentration, that we have the several issues anyway. Next, please. This conventional approach is good, just to just interpret easily. We just everybody can understand what it is, and but issues in this case, if we have the TDM based on 1 or 2 sampling, is not just the easy for the routine use, or not cannot just estimate the AUC with using this method. But for this one, we need something alternative. As I told you, something the modeling, PK, PK or PPK model based on with using Bayesian forecasting. But if we can estimate the AUC or Cmax from the model, or we can use the PK and PD index if we also having the MIC data. This is expected to have the better prediction for efficacy, but not as I told you. And we need something other, the experiment, like the 105 something infectious model experiment, or just in the clinical isolate, or just almost limited data for the MIC from the clinical samples, clinical setting. So therefore, this is still just in the interesting target, but not popularly used in the clinical setting for the TDM. Next, please. This is the Hollow-5 system to just the estimate something such MIC or based on the AUC of MIC for targeting something the response. And in this case, therefore, we do use this in this experiment. We usually use the pre-drug concentration, not the total. Therefore, in the PTPD target, the AUC is F AUC is free drug concentration AUC we use. Next, please. And this is one example of the study in the PK, the concentration, and the mimic the response, and the coronary forming unit, and also TTP. Next, please. So this is a real just the MIC data collected from the clinical data set in the Korean patient. I'll not just talk because time is already over. And the MIC is being Korean, and still the MIC data is not popularly just collected in the clinical setting. Usually we do just a DST as you do. So therefore MIC is just collected from our specific clinical study. So from this, we just got still there are some cases, some samples that are mismatched in the DST data and or this MIC based the crop range. Next, please. Yeah. So whatever we do have still there is something gap in the therapeutic target with AUC and or the population AUC. This is although we just measure why there is something the gap is mainly because we do just this the population data come from the clinical setting. The lower therapeutic target is just come from the Hollow-5 system. So therefore something different. So we need something the solution to just make this in the future as well for us to go use together. Next, correlate. Next. Okay, and the Cmax data is the same issues. Next, please. Okay, and this last slide, in addition to this, in sometimes in the anti-TB medication, there is something the known pharmacogenetic biomarker that are correlated to something toxicity or some response, but not just the outcome, but the PK, the drug concentration. So therefore, through the collection of those information, we just developed something, the multi-gene genotyping kit with using the snapshot methodology for those, the main well-known or representative, the genes and the genetic parent to associated, response, such as the toxicity and the clinical response. This is something the chromatogram, dendrogram for the electrophoregram for the snapshot the study. Next, please. Okay. And we do just some compare each validation for the validation. Next. Okay, this is in the last slide. Anyway, the TDM, although I'm just saying that, I think that almost all clinicians, what is the TDM, or you might be involved with the TDM. TDM in the precision medicine, it's not very old principle. It's very popular and has been used for long years in the clinical pharmacology. And used for the mainly in the infectious disease, but used to use for some other cardiovascular, neuropsychiatry, and many others. But the infectious disease and this, the consensual control assessment, such as TDM, is will be very helpful. That is really one good tool for us to do precision medicine as. Although the previous president in the U.S. Barack Obama initiated precision medicine, but it is not quite a new concept. That just concept, just including TDM as one of the component for us to do in the precision dosing as a part of the precision medicine. This is my last slide. Thank you for your attention. Please next, yes. Let me introduce my colleague, the Professor Young-Sun Cho, who is SI in the project, who I just mainly contributed to those projects, and also thank you to the many clinical trials in Korea and Indonesia and some other country in each day just collaborate with us. Thank you.
Thanks very much, Professor Shin. Congratulations on everything you've achieved. It's quite humbling when we're just starting out and then we hear this talk and you've got nearly 4,000 TB patients with all their data collected. That's obviously a huge amount of work. How many years did it take to get that many patients?
Yeah, it is almost a 7-year project, but we already have 6 years. But what I can tell is, while we are starting, we just need the design and the collection, just to contact the many sites. So therefore, the collection period, I can say 5 years. But unfortunately, while we are doing this project, there was COVID, the pandemic in the world. There was a big disaster to us. For us to do this, because most of our colleague the scientists, respiratory specialists who are just seeing the TB patient, they are just just waste a lot of time exhausted from the management of COVID. So therefore, it is a kind of something how I can say period in which we cannot just make the rapid progress. So. Therefore, I can say we just spent almost maybe 4 years fully, but as you said, it is really big jobs, and we do have the many steps, the many all those, and as well, in each side we do have also one the PM who are just involved in this project as the assist of the clinician professors, and they just collect many information, and we also just have something the QC for the data. This is a really big job. So what I can tell you is anyway the data we collect is qualified and you're well filtered the data. So that's the reason you could get this the following the research such as modeling.
So in Australia, for the NAT2 genotype for isoniazid, nowhere in Australia does that test, unfortunately, and you do it on everybody. So it's we have to send it to America. So yeah, very jealous of everything you've achieved, but it's all come from the hard work of you and your team. So thanks very much, Professor Shin. In the interest of time, I'll move on, but I thank you for joining us from Korea, and you're welcome to stay if you wish for the rest of the talk.